So you can allow people to come in so that I don't have to keep interrupting. And I believe you're in the same area with Samuel.
Yes.
Is that okay? Then I need somebody who is not residing in the same area. Somebody else, please raise your hand who is not residing with Samuel or Nicholas, so that in case of power blackout, you have a second person doing the recording. Between these molecules, and because they are pulling towards each other, there is a tendency, if this molecule is pulling toward this and also pulling toward this, the end result is that it will pull towards the inside. Now, if there is nothing in this area, say it is a vacuum, and you can see that this water film is attached to the wall is the white part. Assume the wall is a balloon, and the water is attached to the balloon. If there is this tendency to try to come in, remember this place, in case it is a vacuum, this will pull in and there will be a tendency of the whole of this balloon to collapse, to come in the middle. If the water film is not attached to the wall, you'll find that it will separate very easily and form a ball, trying to come to the lowest possible area. This is because of the water or surface tension. The surface tension is the interface between this part and this part, the water region and the air, if at all there is. Now, for you to prevent this from collapsing, from coming down to the middle, you have to apply an equal pressure to oppose that force. Now, from the law of Laplace, we say that the radius, if the radius is small, you will find that the tension tends to be very high, because the pressure here that has to oppose that will be equal twice the surface tension divided by the radius. So if the radius is very small, twice the tension will be very high. And for that reason, you really need to put a lot of pressure against that water film to prevent it from falling down to the middle. On the other hand, if the pressure is high, then that high pressure, if it exceeds the 2 times over the radius, you'll find that you're going to inflate this area because you're putting a lot of pressure to oppose the surface tension. Okay, so you'll end up making this volume bigger. And this is what happens normally during inhalation. You create a lot of very low pressure. And because of that, a lot of air is coming in, and that air will end up causing the inflation, and the thing becomes bigger. So now that you can relate the size collapse of an alveoli, this assuming this is one alveolus. Collapse of this alveolus is related to the amount of pressure that you can oppose. So if it is small, you require a lot of pressure to keep it open. If it is large, you need to just add a little pressure just to keep it open. Otherwise, that pressure is already opposing. The size is already opposing. And that is what you'll be explained to in this. So here is the formula. Pressure is equal to 2 times the surface tension over the radius. So if I were to blow this balloon, I would require more pressure here to keep this balloon from falling down, from collapsing. Whereas I don't require a lot of pressure here because the radius is bigger. And as you saw in that demonstration I gave you last time, if these 2 airways were connected and you are to allow these 2 to interact, there will be a tendency of this air inside this alveolar to move up and go back to the one which requires less pressure to stay open. Now, when this happens, you'll end up with collapse of the smaller vessels. The collapse of the smaller alveoli, so that you can inflate or ventilate the larger alveoli. But this is not what happens. We are going to see the role of the surfactant. Further, when you add, in this case, like I told you, just watch the video. It is going to show you what is happening. So at this point, it will tell you about the role of surfactant. Now, in your alveoli, in your body, what your body has tried to do is to try and equate the amount of air coming here to the amount of air coming here. Because definitely, if you don't do anything and air is flowing in from the trachea, bronchioles, and all that, when it reaches here, because it will have to force itself against this high pressure of air trying to come out, it will find it easier to go where there is less opposition, and therefore it will just go straight to the large alveoli. Are we together on that?
Yes.
Yes, before I go to the next part, I need you to understand that part. If you're inhaling and you have small alveoli and you have large alveoli, air will find it easier to go to the larger ones, as you saw in the demonstration of the two balloons. And you'll find that even the one from the smaller ones will try to go to the larger one. Now, this is where the role of the surfactant comes in. What your body tries to do is to produce surfactant so as to reduce this surface tension. So when you put surfactant around here and around here, you make it easier to inflate these two alveoli. So I know I gave a question in the exam, in the quiz too. And in that question, I had 4 choices. And there's only one student who raised the hand to say all the choices were correct. You are supposed to choose which one was wrong. And all of them were correct. So there was no valid choice. For that reason, I will not mark that answer. I will not mark that particular question. So everything will be out of 49. Now, what I wanted to tell you is that with the surfactant, it will tend to reduce the surface tension. So when you reduce the numerator, what you're trying to do is to increase this, and therefore, because you have reduced this surface tension, you find that even the pressure, if you have a small radius, remember if you had a small radius, you needed a lot of pressure. So when you reduce this and you remain with the radius as the same, what you are going to do is to increase this value. And therefore, sorry, you are reducing the value because the radius still remains the same, but then you reduce this and therefore the value becomes, the ratio will become smaller. So what will happen when you do that, the amount of pressure that you need to put here will be very low because you have reduced the tension. So if you reduce this to a very, very small level, then you find this fraction becomes very small. And if it becomes very small, the amount of pressure you require to put in so that you keep it inflated will be very low. For that reason, the opposing force, remember the pressure you need to keep the balloon open is the opposing force, and it is opposing the air that is coming in. I hope we are together on that. So we do have air coming in here, but there is the tendency of this pressure to oppose. So let's put it in blue. So this pressure, which is in blue, is trying to oppose the incoming air. So when the radius is small, we have a lot of pressure here. And because there is a high pressure, incoming air will opt to go there. Okay. But now, when you reduce the surface tension and you keep the radius the same, so what you're doing is you're reducing this value. And when you reduce this value, it means you are removing that opposing pressure. And when you remove this one, when you remove this opposing pressure, you find that the amount that you require to go in here is very small. So here you just require a small amount of pressure to oppose the incoming one. For that reason, as the air comes in here, it doesn't have a lot of high opposition, and therefore it will easily go to this alveoli and it will inflate. Okay. On the other hand, with this large alveoli, it will also produce some surfactant. But you find that depending on the size of the alveoli, the amount of surfactant that you produce will not be the same. Smaller alveoli will tend to produce, remember we have the type 2 cells. And you can imagine if the percentage of type 2 cells is the same in the small and the large alveoli. When these produce surfactant, there will be a very large layer of surfactant. And it will spread out very easily, and therefore it will go in between the water molecules very easily. So when the air comes in, this small alveoli will become inflated to a very large extent. Why? Because you had this thick layer of surfactant. This is as opposed to if we have the same number of type 2 cells in the large alveoli, and they are producing the same amount of surfactant, you find that the layer of surfactant here is very thin. And because it is very thin, it doesn't give compliance. It doesn't add the compliance to this large alveoli as much as it adds to this one. So when you inflate, the inflation here will be very small. It will be marginal. So now, at the end of the day, when you inflate these two, these have a lot of surfactant, they give a large inflation. These have very minimal surfactant, a very minimal inflation. So the amount of air coming in, there will be more going to the smaller vessel, to the smaller alveoli, and less going to the larger alveoli. Are we together on that part? Silence means some people are lost. How many are lost? Am I audible? Can you hear me?
Yes. Yes, we can hear you.
So you are either with me or you are not. I want to see people that I have lost, because the next part we are going to do the alveolar efficiency. And efficiency looks at that rate of exchange here and what happens at the vessels here. Now, if you don't understand this part, go to the next part, you'll be so lost. So I need to know, have you understood that part? The smaller radius will expand more. The reason it expands more is because it has more surfactant production. More surfactants means it increases the compliance more. And therefore, when you're inflating the lungs, these will easily expand more than the large ones. Because the large ones have very little surfactant, so their rate of expansion is smaller. So I'll ask the same question. Have you captured that?
Yes.
Okay.
Yes.
So Asna, I hope you are saying that you have captured, or you have a question.
Yeah, I have a question.
Okay,
I want
let's
to
hear.
ask for the larger alveoli. Where does it have less surfactant?
So if you look at, remember all these alveoli, as they are multiplying, they have the same amount of, shall we say, we have the the two types of cells, type 1, type 2. So what I'm trying to imagine, if you have a small balloon, yeah, and the amount of surfactant you can produce in this is the same as the one you can produce in this. Why? Because you have the same number of cells. If you have 50 cells here, and you have 50 cells here, type 2 cells, each of them will produce the same amount because they have the same rate of production. But the amount that is produced here, because of the small radius, will prevent a thicker layer of surfactant. But because this one has to spread over a big area, the layer will be thinner. If it is thinner, it means that it will not be able to increase compliance as much as this one, because for every given volume of water or molecules of water, you have more surfactant infiltrating as compared to this one. Does that make sense, Asna?
Yes, it does. Thank you.
Okay, so I hope even the others are together. So remember, the larger ones produce less surfactant, the layer is thinner, compliance is less. The smaller one, they'll produce more surfactant, the layer is thicker, compliance is greater. That is the point I wanted you to watch. Now, like I said, today's topic are some of the things I will ask a lot about. So I will take you to the end of the video here. Oh dear. It has decided to go to advertisement. So I will skip that one for now. So what I will ask you to do, towards the end, it's talking more about the role of surfactant in newborns and how it causes, lack of it causes respiratory distress. So if you have respiratory distress, you find that the lungs, and this is simply because they have not matured to start producing surfactant. So if you deliver these children before they can produce surfactant, they will enlarge their alveoli, and therefore, if they don't enlarge their alveoli, they are not able to ventilate their lungs. They end up suffering from respiratory distress. Secondly, because they cannot expand and there is a lot of air pressure, at times you rip the tissue. You rip the alveolar tissue and you end up having bleeding and fluid buildup, which actually makes the respiratory distress worse. So I need you to watch that video because some of the questions I will ask will be related to that. So now let me go back to my topic today. I said that I want to discuss efficiency. And the reason I was showing you that is because when it comes to the expansion, it will determine the level of efficiency that we have. So So today, this topic will sound like a lot of repeating, but what we are having here is that you need to understand these concepts so that you can understand alveolar efficiency. I will take you back to the gas laws. These are the ones that they allow you to predict the movement and direction of flow of air during inhalation and exhalation. And we say that this inhalation exhalation is actually we talk of pulmonary ventilation. Then you remember the pulmonary ventilation captures two concepts. It has two parts. Pulmonary is for the whole lung, but we have alveolar efficiency and the dead space volume. So for you to know how much ventilation you have in the lung, you have to take those two into account. Number two, we go to Dalton's law of partial pressure, which will tell you now the percentages of air that is able to pass through and go and exchange between the air compartment and the blood compartment. So that one is very important because the proportions of gas in the air will be the same that is going into the blood. However, it brings the other component of diffusion. And this is explained by Henry's law. Now, Henry's law is looking at those partial pressures in the gas compartment and the ones that are dissolved. But for you to account for the ones that are dissolved, you have to take into account the diffusion coefficients. Now, this diffusion coefficients is different for the different gases. So if it is the same compartment, air and water, for all these different gases that are in the atmosphere and that are dissolved, the only thing that will cause a difference in diffusion will be, according to Henry's law, will be the partial pressures in the two chambers, and secondly, the diffusion coefficient for each gas. So for instance, we know that carbon dioxide dissolves more than oxygen, and oxygen dissolves more than nitrogen. So you can actually relate that the amount that is being able to pass through, we have more of carbon dioxide, more of oxygen, which are dissolved than the nitrogen. But now once you have them dissolved, they interact with so many things. So when I have, what I have here in summary is that Henry's and Dalton's law, they predict about the inhaled air, how much will be dissolved. But there is also another component. So this is just a summary, and it is a recap of what we have done. Henry's law, which is looking at partial pressure in the chamber versus the concentration of the dissolved gas and the relationship with the solubility coefficient. So I'm going to ask you to look at this slide slowly because I'm trying to brush through it. We had already done it. But what I want to say is if you increase the solubility coefficient, then what you're going to end up doing is when you divide the two, you'll find that the partial pressure will be much affected because there's that, what is it called? Inverse relationship. Now, the concentration of the dissolved gases, if you are to rearrange the fraction, you find that the amount that is dissolved is dependent on the partial pressure. So if the partial pressure is low and you multiply by a high solubility coefficient, then that concentration will be high. So just look at that relationship and you can relate to Henry's law. Now there's the third component that will determine how much of each gas will go. So Henry's law will tell you the amount for individual gases. But now this one brings into account how much is able to move from the gaseous compartment to the liquid compartment. So the determinants are the rates of diffusion. This will be determined by the surface area, and that's why you have a lot of alveoli. It will be determined by the concentration gradient between the two chambers, the alveoli versus inside the blood. And thirdly, the thickness of the membrane. So if you remember, every time I tell you, if you change the thickness, for instance, if you increase the fluid compartment between the gas and the blood, say in case of you have pneumonia and you have a thick layer of mucus, it means you're increasing the thickness through which the gas has to diffuse. So for that reason, according to Fick's law, you're going to find once you increase the thickness, you're going to reduce the amount that flows. If you increase the surface area, you increase the rate at which it flows. If you increase concentration gradient, similarly, you increase the rate at which it flows. So those are the most important laws that we are going to think about, which will end up with this again summary slide. We had done it in the last lecture, where we have the inspired air and the partial pressures and pressures in the tissues, in the venous blood. So this was the arterial blood, this is venous, and then we have the expired. So keep those values in mind because they keep changing, and they will change according to those laws that we have just described. So this diagram, again, I think it was among the last ones we had discussed in class, where I was showing you the layers through which they have to pass through. So if you increase the surface area, you are increasing the flow. If you increase the thickness through which the gas has to pass, you reduce the amount that is available to the blood or that is able to diffuse or exchange. So if you increase the pressure, then again, that takes into account the concentration gradient and therefore movement of the air. I don't know whether my summary part has been so fast before now I come to this part. Are we in agreement up to that point, or do we have a question so that I introduce now my new topic? Are we in agreement, or do I have a question? And please don't keep quiet. I'll start calling names just to make sure people are following. Sabata. Sabata, are you there?
Yes, I'm here.
And you're able to follow?
Yes, I'm following.
And you're not agreeing or disagreeing with me. Mustafa Eden.
Yes, I'm around.
You are around, following or
Yeah.
asking a question?
No, I've been following.
Okay. So far, remember, this is recap. Recap. We have already discussed all those in the different lectures. The reason I brought them is because today's topic looks at alveolar ventilation efficiency. So it is saying how much ventilation do we have going into an alveolus? And then how much blood is coming to that alveoli? If we have so much blood, remember we talked about the countercurrent exchange. We have this amount of air coming in, and we have blood flowing in the opposite direction. By the time each of these leave those two compartments, the partial pressure should be equal, so that the one in the blood should have equilibrated to the partial pressure that was in the air. The one, when you have the amount of air that was low, say, for example, if we had low carbon dioxide in the air chamber, by the time the air is moving out, it will have equilibrated with the amount of air that is coming, with the amount of air that has been brought with by the venous blood. So that is simply the countercurrent exchange. And from here, if I can just draw roughly an alveolus, and then I have a capillary. coming here. So you inhale, and when you inhale, blood is moving from one direction to the other. So it will start exchanging at this point. At this venous blood, very high in carbon dioxide, low in oxygen. So it will keep exchanging, and by the time it is leaving into the venous capillary network, it will have equilibrated, and the amount of carbon dioxide here will be equivalent to what is here. And now, when it comes to the oxygen, again, it will have exchange to the same level. So this is what we are calling the countercurrent, whereby the flow, you can assume that the flow is coming this way and going that way. Or you can bring it the other way around and going this way. But what happens is that by the time this blood is leaving, they will have enough exchange to the extent that the air moving out will have equalized with the blood that is going in the venous circulation. Now, having known that, we have looked at ventilation and perfusion, and now I'm going to talk about perfusion. Perfusion is the amount of blood that is flowing to the tissues. And how can you determine perfusion? Because if we are talking of the lung is supplied by blood, supplied with blood from the heart. So we have the heart here, and through the pulmonary artery, it will go into the left and the right lung. So the amount of blood that is pumped out with every heartbeat, we call that the stroke volume. Every heartbeat gives a volume of blood that is kicked out. Now, if we take that volume and you multiply by the number of heartbeats, it will give you what we call the cardiac output. And this cardiac output is the one that goes to the lungs. So when we talk of perfusion, we are looking at what is the cardiac output that is delivered to the lungs. Are we together on that point?
Yes.
Okay.
Yes.
So now it means here in this particular formula I have here, this was just trying to show you how to calculate the alveolar ventilation. And it takes into account respiratory rate, tidal volume, dead space volume. So I remember we did that in the last slide. Now, when we are talking of efficiency, we have alveolar ventilation is the one that is determining how much air is available for exchange. So if the alveolar ventilation is high, then you have a lot of air that is available. If it is low, it is not. Now remember, the dead space volume usually doesn't change much. And if you recall, we said that is about 150 ml per breath. So per respiratory cycle, it's about 150 ml. Now, if you take into account, generally, respiratory rate ranges between 12 to 15, let's say to 20 breaths per minute. So you can easily calculate how much air is moving in and out per minute, because this rate is given breaths per minute. This is a volume. So when you multiply the two, it will give you the total alveolar ventilation, having removed the dead space volume, which is roughly about that value. Now, this is where I remember mentioning in class. When you have low oxygenation in your blood, you can choose to increase the heart rate, not the heart rate, the respiratory rate, so that you start panting. But when you start panting, you are just simply increasing this rate by a given value. So in this case, they are giving an example. What if you are to double it? If you double the respiratory rate, it seems you just simply double the alveolar ventilation. And when you double it, this is the value you get. Now compare this to if you don't double the respiratory rate, but instead you double the tidal volume. So the tidal volume, one breath gives you 350 mLs. So 350, if you multiply it by the respiratory rate, it will give you roughly about this. Now, if you double the tidal volume, it means in one breath you have about 700 mLs. If you take that instead of 350, if you multiply the 700 mLs times the respiratory rate, if it remains at the same, you can see what the alveolar ventilation comes to. So, which one would be easier? You are low in oxygenation, you want to increase alveolar efficiency. What are you going to increase? Respiratory rate or the depth? Because when you increase the tidal volume, it means you are increasing the depth of breathing. You are taking in more air. This is what happens when you yawn. So all you remind yourself is, as you sit down in class, your rate is very low, you are building up carbon dioxide, and you start feeling you are stressed due to the acidity. The next thing, unconsciously, you're going to take a deep breath. We'll gasp in a lot of air, and you're going to restore your air because you have increased efficiency. So this is where I start calling people. People at the bottom. Prince Moses, are you okay? No microphone. Wakagio Ashira.
Yes.
Did you capture that concept?
Yeah, I'm following.
Okay. So now I will go now to the next step. So I have quite a number of concepts that I have brought in here and there. They are building up. I'm trying to build up to where we are going to go today. So here I have already discussed increased alveolar efficiency. I have also talked about reflux law, so I will not go through this again. Instead of using the slides, I used the video. So again, I implore you to go and watch the video. So the whole idea of surface tension, it prevents, when you put surfactant, it will tend to prevent collapse of the small alveoli when you have unequal alveoli. And they also talk about edema, but I think I'll skip the edema for now. That one I'll come to later if we have time. So from the video discussion I have just given you, you are trying to match ventilation with compliance when we have an equal sized alveoli. So when we have small alveoli and large alveoli, you try to match ventilation by increasing surfactant in the smaller alveoli so that you increase compliance, which will require less pressure to inflate. And therefore, for every one inhalation, when the pleural cavity or when the pleural pressure pulls away the lung, it is pulling away both the large and the small alveoli. But because the small ones have a greater compliance, they are going to increase in volume much more than the larger ones. Why? Because the larger ones have less surfactant and less compliance. So at the end of inhalation, these two alveoli, smaller and larger, will be relatively the same size, only the difference being with the small alveoli, it has moved from one volume to a very large volume. With the large alveoli, it will have moved from a large volume to just a small increase. So at the end of the inhalation, you can see they are almost the same size, but the margin of increase here is smaller than this one. And therefore, this has brought in more fresh air. And this more fresh air is more available for ventilation or for gaseous exchange. So in the smaller alveoli, we have greater ventilation. Have I confused anybody up to there? In the smaller alveoli, we will have greater ventilation. So I think before I come to this, let me explain this diagram. Then I'll go to that slide. Have you done anatomy of the lung?
No.
Was that no or yes?
No.
Okay. So generally, when you look at the lungs, towards the apex, we have very large alveoli. But as you come down to the base, they become smaller and smaller. Okay. Now, that is when it comes to the lumen of the alveoli, and this happens to both sides. So the size of the alveoli decreases as you go down to the base. Now, secondly, if you bring in air, it means these top alveoli, they have large airspace, but from Laplace's law, you find that their ventilation will not be as high as these ones at the base. Why? Because these ones will have more surfactant, their inflation will be more. Excuse me. Now, the other component you have to think about when you look at the lungs, and that's why I asked you whether you've done the anatomy. The heart is located roughly in this middle to lower half of the lung. So when we have the pulmonary arteries, they are roughly just around the mid portion of the lung. Now these arteries have to pump blood all the way to the apex and all the way to the apex. But at the same time, we have blood that is going down to the base. Okay? And at this same time, we have blood that is just in the middle portion. Now, for combined reasons, if you have an upright person, if you stand upright, there is always the effect of gravity. Gravity will tend to pull the fluids to the lower parts. So you find that the lower areas will have more fluid than the top areas. And that could actually explain the sizes of the alveoli. So you find that the interstitial spaces and all that fluid will tend to try and settle to the bottom, and therefore the alveoli are able to become larger as compared to these other ones. Now, when it comes to blood, blood is also a fluid. So as you pump it, it has an equal pressure at the middle region. But it will find it easier to go to the base area than to pump upwards against gravity. And therefore, if you look at the amount of blood that is going to the apex, it's very low. Why? Because of the combined effect of the pump plus the gravity, it will end up perfusing these smaller alveoli. For that reason, before I erase here, do we have any question on that? Two components. Alveolar size decreases as you go from apex to the base. That is one thing. Number two, the blood perfusion and pressure will increase as you go to the base as compared to the apex, because it is bringing in the component of gravity. Therefore, if you have more blood going to the base, we say that there is greater perfusion. So with that, if you have no question, I will erase what I have written here and you'll see the summary you have in this diagram. So at the apex, we have low ventilation. Who can remind me why we have low ventilation at the apex as compared to the base? Because that has been the topic of the previous slide. Yes, Lucy?
So at the apex, we have the large alveoli.
Yeah.
Yeah, so the large alveoli has a lesser surfactant,
Yeah.
and therefore the ventilation is low.
Yeah, so when they start to expand, they won't expand as much as the small alveoli. So the ventilation will be greater at the base. On the left side, we look at perfusion. We saw that we have low perfusion and we have high perfusion. But now when you compare the perfusion to the ventilation, you find that this one is so much lower than the ventilation. When you look at perfusion, it's so much greater than the ventilation. So the total combined effect, if you are to look at the ratio of ventilation to perfusion, remember, we have low ventilation here, but we didn't have, high ventilation, which is one arrow, but we also have a higher perfusion here. Okay. So if you look at that ratio, we have a small increase, but a higher increase in perfusion. So remember, when we start looking at perfusion, we are looking at the ratio of ventilation to that of perfusion, where ventilation is represented by V and perfusion is represented by Q. Now, when you come to this part, you will find the opposite. We will find that ventilation is low, but the perfusion is even lower, very small. So this is low, but this is even lower. So you'll find here the numerator will be high. If the numerator is high, it means that this has a greater dQ. Okay. In this case, the numerator will be smaller than the denominator, which means it will have this one will have a very high dQ, and this one will have a low dQ. For the simple reason because the numerator is smaller than the denominator. So it will have a low dQ ratio. I hope I have not lost people here. I'll go slowly. You understood why ventilation is higher in the base as compared to the apex. And now you understood why perfusion is even much higher at the base as compared to the apex. Now, when you want to look at alveolar ventilation efficiency, you look at VQ ratio. What is the ratio of incoming air to the perfusion of the blood? So the incoming air has, in this case, a low ventilation, but the blood coming in is even lower. So generally, the numerator is low, but the denominator is low. When we put them as a ratio, the V will be much higher than the Q. Anybody lost at that point before I go to the next one? Okay, so when we go to the lower part, this is at the base. At the base, we have very high perfusion and we have high ventilation. But when you compare the two, perfusion is much greater than ventilation. We have more blood vessels. We have more blood flow going to these areas. So the perfusion here is much higher than ventilation. So if you look at the VQ, it is low. So if you look at the ratio of VQ coming from the apex, going all the way to the base, you have a decreasing ratio of VQ. Are we together on that point?
Yes.
Okay. Akelo, you have raised your hand. I don't know whether
Yes.
you have a question.
I do. We were talking about ventilation. You said there's low ventilation, and then you said oxygen will also have. I didn't get that point concerning the V over Q on the ventilation part of the apex.
Okay, in the apex, the ventilation is low. Why is it low? Because we have large alveoli. With large alveoli, one of the things we have is low surfactants and low expansion. Okay?
Yes.
In the base, we have small alveoli, more surfactant, more expansion. So at the end of inhalation, we have more air coming in. This one, at the end of inhalation, we have very low amount of air that has moved in. Okay? So
Yes.
in this case, we say low ventilation. In this one, we say high ventilation.
Thank
So
you.
does that, okay. So, and remember, ventilation is the amount of air coming in and out and the availability of oxygen for diffusing. So if there is
You
low
also
ventilation,
mentioned. Sorry,
Go
you
ahead.
mentioned something about blood.
Okay, now for the blood, remember we said that the heart is located at this area, but the blood is coming from the arteries. We have the pulmonary artery that will take blood to these areas. They take it around the middle part of the lung. Now, when you have blood, it's a liquid, and the liquid responds to gravity. So there is the force that is trying to pull the liquid downwards. Now, there is the pumping, there is the force coming from the heart. So you have the heart pressure, and now that blood pressure is combined with gravity, so most of the blood will be going down to the base. So we'll have blood vessels, blood capillaries, and remember, all these tiny alveoli all have very many tiny capillaries. So the volume that is actually going here is much, much more. Volume of blood, because you have more capillaries, you have more pumping, and you have more effect of gravity. So the amount of perfusion here is double. And that's what I go to the left side. The red is representing blood flow. This one will be much, much more than at the apex. Why? Because the apex is pumping against gravity. So you're trying to use blood pressure, but the pressure is moving away from the gravitational force. So the force is trying to pull it down, but you're moving against it. So there will be less perfusion here. Okay? The alveoli are larger. And therefore, remember, we say that to increase surface area, you need smaller components. So now we have very large alveoli, so even the capillaries that are surrounding them will be fewer. If they are fewer, the amount of blood going there is less. Now we have less perfusion because we have less blood, we have less pressure. Okay? So this one, the perfusion will be much more. So that explains perfusion. But now, for you to understand ventilation and perfusion, you have to bring the two in together. What is V over Q? It's a fraction. It's a ratio. Take ventilation and use it as the denominator here. And from the arrows you have here, you have one arrow as opposed to two. It means the denominator is higher. If the denominator is higher, the fraction is small. Sorry, if the denominator is larger, it means the fraction is tinier. Okay? So when you come to the VQ, where we have low ventilation and you have low perfusion, it means that the VQ will be small. However, if I go to this part and I take the ventilation, which is high, and I divide it by the two arrows, which are even higher, so you'll find that this level is much lower. The numerator will be much lower than the denominator. And for that reason, this one, since it is so low, there is a decreasing VQ ratio. Okay? A decreasing VQ ratio, just because the denominator here is higher as compared to the apex. Okay, Alice?
Yes, thank you.
Okay, so with that in mind, I have this slide. I skipped it because it has a lot of writing. Now, as you can see, the bottom right here shows you that there is audio to this. You have an audio listening. So what I have done is I have produced a PowerPoint recording, which is explaining all these components in details. And they have my voice that is actually explaining each of the slides. I will post the audio recording of the slides, not this particular lecture that I have today. This is previously what I had made of the alveolar ventilation efficiency. Then if you don't understand any component, you can listen to the audio, just go straight to the slide and play that slide and it will give you the narration and you'll be able to understand. So to look at when you're trying to match alveolar airflow with the blood airflow, we are looking at what is a VQ ratio, where V represents ventilation and Q represents perfusion. So if I were to address ventilation alone, ventilation is the amount of air that is available for gaseous exchange, which means it is the alveolar ventilation. We calculated alveolar ventilation by subtracting the dead space volume from the total pulmonary ventilation. And we said when you do that, it comes to about 4 liters. And this was in the previous slide where we worked it out. It comes to roughly about 4 liters. Then we address it. Since we know the normal ventilation, that is at quiet rest, a tidal volume of 500 ml. So you are at quiet breathing. If at quiet breathing, we look at your heart rate, it is about 72 beats per minute. So now we have moved away from the lungs. We have moved away from ventilation and we are focusing on blood flow. How much blood is available to the lungs? We are going to look at the amount that is being pumped by the heart. And the amount that is being pumped by the heart, it all ends up in the lungs. And the amount that is being pumped in each heartbeat is what we call the stroke volume. So the stroke volume for every heartbeat ends up being directed to the lungs through the pulmonary artery. Now, for you to know the total amount of blood that will flood the lungs in a minute, you have to look at how many beats of the heart did we have in that 1 minute. And that is the heart rate. So you need to multiply, you need to multiply the heart rate by the stroke volume so that you can get, just give me 1 minute, please, so that you can get the cardiac output. Stroke volume times heart rate will give you cardiac output. Just give me, oh.
Excuse me.
Hello. So I'm being called for another meeting. So I need to wind up. But this lecture actually, that's why I made the recording so that you can be able to see what happens. So what I was trying to show you is this diagram whereby at the apex we have large alveoli but low perfusion. So if you start looking at the VQ, you find that the VQ is decreasing as we go down to the base. Now, the last slide I wanted us to discuss, this one is giving a summary of the same, but I wanted to show you what happens in this case when you start changing the VQ ratio. So if you alter ventilation or you alter perfusion, you're going to have what we call a mismatch, a VQ mismatch. If you want to look at the VQ in general, you take a summary of all these areas, the zone 1, zone 2, zone 3. Add the VQs because the VQ at the apex is roughly around 1. If you go to the middle portion, it starts to reduce maybe to around 0.8. When you go to the base, it's roughly around 0.63. So when you add them up and you divide by all those zones, you find that generally the VQ ratio is about 0.8. So the normal VQ ratio is about 0.8, but it is not equal in all the portions of the lungs. It decreases as you go down, it's higher at the apex. So what I wanted to show you is in the individual alveoli, what would happen in a small alveoli if you are to decrease ventilation or if you are to decrease perfusion? What can cause increase in ventilation? So if you lower the amount of blood which is supplying that particular alveolus, say for example, if you have a blockage of an artery or a blockage of a capillary, the blood flow to that particular alveolus will become low. So if this becomes low, you find that the ventilation becomes very large, and we call that wasted ventilation. There is a VQ mismatch, and therefore the exchange is not perfect. Now, on the other hand, if the blood flow was okay, but you had a blockage of the incoming air, say for example, you have a bronchitis or you have asthma, which decreases the amount of air that is available to the lungs, you'll find that what you'll have is low ventilation but high perfusion. Again, there is a mismatch. So the rate of exchange will be minimized. So you find what the the lungs tend to do. To prevent that, and to prevent what we said in Laplace law, so that you end up having air going to where it is not supposed to, what the body will do is to alter the diameter of the bronchioles and alter the diameter of the blood supplying them arterioles. So if it alters this, it can vasodilate where we have a blockage, or it can vasodilate where we have an obstruction. So this is what we are calling trying to match, trying to correct. And I think that is the last thing I wanted to discuss before I go to gas transport. I will capture it more in class as we meet. So here we say that matching alveolar airflow with blood flow, the pulmonary vessels can constrict or vasodilate. They constrict where we have low oxygen. So if we have that low oxygen here, let's see. In this case, when we have low oxygen coming in, these blood vessels will vasoconstrict. The ones with high oxygen, they are going to vasodilate so that there is more blood going to where there is more oxygen. So that will sort of cause a matching effect. If, on the other hand, we have low oxygen coming in the ventilation component, high oxygen but low blood, then the bronchioles will do the opposite. The bronchioles will vasoconstrict so that they allow less air to flow in, so that there is a matching of these two. And I think that is the last thing I will leave you at, so that we can capture it in class. I will review it in class, then you can see. Remember, these topics will take 10 to 15% of your marks. You really need to understand them. If you have a question, you let me know. Okay, please type your names in the chat as I open and join my other meeting. So please quickly type your names in the chats. You're 37. I need 37 names quickly so that I can log you out. I start my other meeting. Thirty-four have typed. Who are the other 3 people? Type quickly. I want to log you out. If I log you out and you have not typed your name, you are absent. One more person, that is me. So I can end the meeting. See you next time.